So I would like to discuss with you a bit on how I think we can study polygenic adaptation with experimental evolution, and I will try to cover some different approaches that we are doing, and curious to hear some feedback from you, what you think about this. So I don't want to spend much time on this, but basically saying that the genetic architecture matters. What is the adaptive response we expect to see at single SNPs? With polygenic adaptation, as you know, we expect very little frequency change at adaptation compared to sweeps, where we see a very pronounced signature that extends in the flanking regions. So there are a lot of methods available for detecting and all this, but that's not what I want to cover. The idea is what I would like to introduce is a concept that we came up with in nomenclature to distinguish polygenic traits that matter for adaptation from polygenic traits, not traits, loci, QTLs that, or loci contributing to a polygenic trait that are detected by GWAS, because there is the idea that with GWAS, there are several reasons why the loci you detect with GWAS cannot contribute to adaptation. The first one is pleiotropy, which I think is a very important one. Another one could be linkage. And the third one is that these loci that you detect with GWAS are just unconditionally deleterious. These are bad alleles, and they can never contribute to adaptation because it's just bad. So there's a lot of reasons, and that's why we think we don't talk about the genetic architecture of polygenic adaptation, but we call it adaptive architecture because this captures those loci that are responsible for the polygenic adaptation. And this is a difference to those ones that you detect with GWAS. So what I would like to tell you how we are doing the characterization. What we do, I will first talk a bit on genetic redundancy, which is a hallmark of polygenic adaptation or polygenic traits in general. How we can go about trying to estimate the number of loci that are contributing to a polygenic trait. And then I will talk about complications that could be pleiotropy, linkage disequilibrium, multiple stressors, and multiple stressors is something that is what if you want to go very simple, this is E by E interaction, and not G by E, what Luisa has introduced, it's E by E. And then I will talk about non-additive effects. So I have two different experimental approaches that I will basically using for different parts of it. And so to make it easier for you to distinguish them, when I talk about experimental evolution with many founders, I'm using red. And when I'm talking about experimental evolution that has only two founded genotypes, I will put blue headers. So this is easier for you to follow, and I don't have to explain it all the time. Or well, this was coincidence. So, so as I said, genetic redundancy is a hallmark of polygenic adaptation. And the way we define it is not that there are more loci that could contribute. This means these loci that when they would acquire a mutation could contribute to the trait. But what we think is this is variation is already segregating in the population that we are analyzing. So this is redundancy. This is our loci that have variation that could contribute to adaptation with this trait. And the important thing is that we have more of this variation that we need basically to achieve a shift in trait optimum, which involves the adaptation. So there is a surplus of variation that we have available, and we don't need it all to get to the trait optimum, to a new trait optimum. And so this is the study from Neda, who will be here next week, that basically is one nice illustration how we can identify genetic redundancy. So we had 10 parallel populations that were involved from the same founder, the same environmental stressor. And what you can see here, this is this grayish or brownish, is a color-friendly colorblind friendly colors, so that you basically see this is the ancestral and the other ones are all the evolved ones in each 10 of the replicates. So you can see they have clearly evolved, and here you can also see that they all evolved, and all the replicates have basically consistently moved in the same direction compared to the ancestor. So we have parallel phenotypic change in the old replicates. They do more or less the same. But if we look at the genetic level, we see that the replicates are behaving differently. This is one of the loci or regions that we have identified. Each of the panels is one of the 10 replicates. And so you can see that they behave very differently. In this replicate, we see a very nice frequency increase. And another replicate, basically nothing is happening. This is Drosophila similans. Different lines are individual SNPs. This is a haplotype block. It's basically, we don't have the resolution. We don't have enough recombination, so we get blocks that are basically moving along. And so each of these lines is a member of the block. They vary in size. They can be megabase. It can be quite big. This is one block I'm looking at, yes. It's one example. She identified roughly 100 blocks that show this redundancy pattern. Okay, good. So you may ask, this is may say this is just a single population. Maybe this is something specific to this population. It was from Florida. So I show you here another graph from a population from South Africa. This is a slightly different way of presenting it. This is a PCA, and each of the colors shows one of the replicates. And then we have different symbols, and the symbols indicate the generation at which we have been sequencing the population. And so you can see that this is the ancestral population, and then you can see that the individual replicates are basically evolving in entirely different directions. So this is a very, basically bottom line is, this is not something specific to this one population we've been studying. You find redundancy also in other Drosophila populations from other founder populations that are quite distinct. No, it's the same. It's the same environment, the same founder, but just replicate populations. So you basically, have replicates. They evolve in parallel in the same environment, but from the same founder. Same founder. Yes, also before. It was the same founder doing different things, having phenotypic convergence, but on the genetic level, they do different things. That's the idea of redundancy. So you can basically reach...
It's the same founder population?
Same founder population.
The distribution across the population?
It's the same founder population. You can do it with fruit flies. It's quite easy. You have lines. You can use them to set up replicate populations. And so they evolve basically in parallel. They get the same phenotype, but do this by using different sets of loci.
I just forgot which is where
What? Okay.
So consistency is your founder population.
It's the same founder population.
How many individuals?
We start between around this experiment is with 400 isofemia lines that are being started. I don't want to confuse you, say 400 lines, and the population size is 1,200. Sorry to confuse you with the numbers. I could spend a long time to explain this, but...
So the idea is that some aspects of the diversity is variable enough between the different founder populations to get this going in such different directions. Why isn't it bouncing all over the place? Why is your name roughly the same? Same selection?
It's the same selection, yes.
Right.
But maybe we put it later. I think it has to do, of course, with drift. This is, you have many loci contributing, and in the initial phase, it matters what drift does to them. Drift can act synergistically for one of the loci, and then this is an advantage and contributes more in this replicate. In the other replicate, drift goes in the opposite direction, and this locus does contribute less, but another locus basically substitutes for this. That's the idea.
But if you look down along these lines, even on the first one, what is the distribution of the number of
No. I'm pretty sure we have all the alleles in all the replicates. Okay, good. Well, let's talk about how we could use it. We haven't. Okay, so I tried to show you that we have redundancy on the genetic level, but we have different levels. And in the end, we have the high-level phenotype that is actually be selected. At the bottom, we have the SNPs that I've been talking about. And now the interesting thing is if you have intermediate phenotypes, we find redundancy on the intermediate phenotypes as well. And this is what I will discuss in the next couple of slides with you. So let's start with gene expression. Do we find redundancy on the level of the gene expression? Gene expression is the phenotype that's closest to the SNPs. And what we did basically 100 generations in a novel hot environment. It's the same population that I showed you for Bargi et al. Here we use 2 replicates and we studied the variance in gene expression that has been very nicely introduced by Luisa. So what we find, we basically look at genes that change in variance but not in mean expression. So what we find is that of these genes that change the variance, there are only 10% of these genes that evolve variance are being shared between the 2 replicates. So this could mean, yeah, variance is difficult to estimate trait. We haven't done 1,000 individuals. This was a relatively moderate sample size. So how can I show whether or not this is just noise or something biologically significant? So we find that basically these genes in the 2 data sets share Go enrichment. So they show, basically I've put you some Go terms there. So they share the same Go terms. And what I think is even more striking is they show the same tissue enrichment. So they basically, these genes are expressed in the same tissue. So we find there are different genes evolving the variance, but they basically do the same thing.
So what are these tissues? I don't know the name of the tissue.
But this is basically midgut.
Midgut. Okay.
This is the midgut. So you have many others, but the midgut was enriched. So basically, this is a summary of this. So we think that in population 1 and population 2, you have sets of genes that are basically contributing to digestion, but the 2 populations use different sets of genes to evolve the digestion. And how is digestion evolving? So we bring up this hypothesis that has to do with the lab environment in which we put the flies, because they have been evolving in a very special environment. Typically, flies live in a very diverse environment, but we bring them to a lab where they live on agar substituted with some sugar and cornmeal. So it's a very, very simple food. And so we expect that basically they are adapting to the new food source in the lab. So what we see basically is that this is the fitness function that we have in the ancestral population. You need to be adapted to very broad diverse sources of food. In the lab, the fitness function changes because you are specialized to a certain type of food, so the fitness function becomes much more narrow, and it of course stays the same over time. And then as a consequence, the phenotype changes. We have a broad distribution. In the wild population, it's still broad when we bring them to the lab, when we start the experiment, and then after 100 generations, the fitness function has pushed the variance to be much narrower than we saw in the ancestral population, and this is the outcome. So
this is a schematic. This is data.
Well, this is schematic. The data, yeah, but the data show this, that we see that the variance is reducing for most of the genes, and that's where I showed you the enrichment. So that's a schematic summary.
But you haven't done the distribution.
No. It would be very.
A lot of information in this.
I don't think we have the data good enough for this, so yeah? Two. No, the replicate, we measure the variance in 2 replicates. No, it's not a pool, it's in the single flies. we measure it in single flies, then you can get the variance among the flies from the one replicate, and you do it variance in the second replicate.
So my question is, how many flies do you have?
It's something 40 or something.
And they're all
No, they're sequenced separately. Otherwise, we wouldn't get a variance. Yeah. Yes. And the ancestral. No, there's no change in mean. There's no change in mean. Only the variance evolves. Well, it's very similar to what Luisa saw. It's just this is evolution. That's the only difference. But sorry, can you say again?
compared to the initial population, you have lost it.
Yes. So, okay, good. So now let's move on the next level, phenotypic level, go from the transcriptome to the metabolome and ask what happens with redundancy on the level of metabolites. Do we find redundancy on the level of metabolites again? It's the same population, it's the same individuals that have, well, not same individuals, the same population in the same replicates that have been studied.
Just like following up on the previous one about the diversity. So if some of the alleles that are being more strongly selected are relatively low frequencies initially, then some of the lines will lose them. I don't know how. Is that consistent with what you're having, which is then determining which direction you're going?
Well, the haplotypes that are not responding, we don't find them to be lost. That's on the genetic level.
No, but I guess I'm wondering the ones that are responding strongly, if they're initially low frequency initially, then they'll be lost in some of the lines.
No, because when we look at the 100 haplotype blocks, we find them in all of the 10 replicates. They're there. They remain just at the low frequency, so they're not lost.
And the haplotype blocks are linked such that they stay a block through the number of generations?
They have been characterized as a block by looking at the frequency trajectory over time. That's the, yeah, how we identify them.
No, I understand, but if I ask you over 60 generations, whatever, and I ask how long blocks are conserved over 60 generations, not chopped up over 60 generations.
Yes.
But those are shorter than your linkage blocks.
No, these are the linkage blocks that are conserved over 60 generations.
And
And there are, we confirmed them by sequencing individuals in the evolved lines and the ancestral lines, and we find them both in the ancestral and in evolved lines. So these haplotype blocks are real, and they exist, and they can be large.
do you have some idea as to why you get this going off in a different direction?
I try to explain it because they're not needed. You have redundancy, and so other loci are doing the job, and those ones don't do the job, so they're not needed.
They all have the same frequency of all the alleles. Then it's pretty deterministic.
It's not.
Yeah. Because the populations are different.
Yeah, the populations are small, and this is.
It's also frequency.
If you would have gigantic populations, then you wouldn't see the differences. I think the point is that we have relatively small populations where you can see this. That's,
So the first few generations make it different.
I think the first few generations are important for triggering these differences. Pardon? Not the phenotypes, only we have the expression, we have the sequence data, yeah? And you could see it in the other plot that they were basically already initially diverging in the star phylogeny, in the star-like pattern on the PCA. So, okay, so what we see on the metabolite level, if you compare it to gene expression to metabolites, we see that the consistency overall is higher over the metabolites than what we see over gene expression. And so one example I give you for the purine metabolism that we looked at, this is basically showing the network that you can see here, so it's a bit difficult to see. So we look at the transcript data, and here the individual replicates are shown here in different colors. And if you have, well, in these blocks, and if they have different shades of the color, between green and red, this shows you the expression intensity. So this shows you the heterogeneity that you can have in gene expression on the different genes that we could measure the expression in this network. And you can see that there's quite some heterogeneity on the gene expression level. But if you look at the metabolites, which are in the circles, these are more difficult to see. They are pretty homogeneous. There's very little heterogeneity among the replicates. Just showing another way to show this, that we find much less heterogeneity on a higher level of the phenotype than we see on the expression, and even more than on the nucleotide level. So there seems to be a hierarchy of traits, and the higher in the hierarchy of the traits you are, the more parallel is your response across the replicates. However, I don't think this explains everything, because it's a very artificial thing. It depends also on the network structure you have, etc. But as a rough rule, you can say more high-level phenotypes will show more parallelism across the replicates. Sorry, what? Yeah. Yeah, it's a full change. You compare the ancestor to the evolved one, and then ask how much do they change. The expression can go up or can go down. That's minus or plus. Okay, so the next question is, what is polygenic, and can we measure it? Short answer is no.
There's a quick question. You check the variance in gene expression, and that could be potentially because it's adapted to reduce the expression of the or because of loss of genetic variation in general. So do you see, if you were to measure the variance of other phenotypes, do you see that all of them?
Most genes don't change their variance.
Of your other higher order phenotypes. I'm just trying to figure out if you could explain the changes in variance just because genetic variation goes down.
If genetic variation would go, if it would be genetic variation, it should affect all the other expression traits as well.
No, but other traits.
Well, we don't have this data. So, but I would expect it for other, and I talk more about the variance changes.
Okay.
So, and I don't think this is the thing. So, one way to try to, where we think we can try to estimate the number of loci is exactly looking at the variance change. And we've been hearing a great deal about from Nick and all this, that basically, if you have a polygenic infinitesimal under the infinitesimal model, basically an adaptation should not result in a change in variance. The question is like, does it apply to our populations as well? Because our populations violate the assumptions to a great extent. We have a lot of LD, as I've been talking about. We have small population sizes, and it's a completely different situation. There's no equilibrium, there's nothing. And that's why we did some computer simulations asking what's happening. If something is neutral, and then we think about different numbers of loci, you can see here in 10, we differentiate between 5, 50, and 1,000 loci. It's a moderate version of an infinitesimal model, but you can see that there's basically no effect on the number of loci that you have with respect to change in variance. Why is variance still changing? And this is, of course, because of drift. We lose some of the alleles in the experiments, so there will be some reduction in variance just because of drift, because we have not incorporated new mutations. Most of the variation is, all the variation is basically that we consider comes from the founded population that has been coming from a much larger population than we have in the lab. That's why we have basically much higher variance in the beginning of the experiment. So what happens now when we have adaptation? If you look at this, this is now in this yellow color. We have, when we have a different number of loci, we can see that the variance has been not very much reduced. Only when we have very few loci, we see a very pronounced reduction in this. So overall, it looks like that we can generalize, even for our population, the idea that the variance is not expected to change very much when we have a highly polygenic architecture. Of course, the problem is what are our assumptions? There are many things we need to think about. What is basically the variance in the fitness function? What is the distribution of the effect sizes? How many generations do we run it? And so I give you an overview, and also how much shift in trait optimum do you have? Do you have a strong shift? Do you have a weak shift, etc.? So you can have different things. And of course, it depends on this. But if you look over it, you see that typically when you have a relatively small number of loci, you can distinguish this very well from the expectation with many loci. So the question is, can we basically say whether our expression changes that we see in our experiment or non-expression changes, the variance change, is this compatible with few loci or with many loci? Mm-hmm.
So that you should never have the selected population match drift, because the real frequency change is comparable, but the negative drift is spoken by selection.
Yeah, Laura. Yes. We're assuming that this is well, we don't know what the ancestral population had. We have no idea about this. So what we only can talk about we can talk about what we think was we have the ancestral phenotype, the ancestral variance, and then we can ask what is our expected shift in trait optimum. And for this, we assume a fitness function, and this fitness function has a certain variance that we're assuming. But yes. This was only set for very, very tiny subset of loci. This was only for a very small subset. This was, I don't know, 1% of the loci that showed this strange pattern. This was 100 loci that showed the change in variance. The other ones didn't have any. So you're right, for these ones we assume a change in fitness function. That's correct. This is the conclusion for this very small subset, that we have a change in fitness function. You don't look convinced. Okay, so what do we see? Of course, our data are not perfect. We have only expression variance at 2 generations, 0 and 100. And this is again for 2 populations. And we look at the genes that are not selected versus selected. So how do we define selected genes versus non-selected? We look at those ones that have basically non-selected are those ones that are not changing in mean, and the selected ones have a significant change in mean. And when we look at the variances, they are basically indistinguishable. So in principle, this suggests that it's not very oligogenic, the expression pattern that's driving the expression pattern that we are seeing. But of course, what is also obvious that you would get a much better resolution if you would have multiple time points at which you would estimate variance. So for getting a much better basically handle on the number of loci that are basically contributing, it would be much better to have a time series over which you measure the variance in gene expression, and then you could get a much better idea on the number of contributing loci. Should also say we have very little power to say anything about an individual locus because the data are so super noisy, the population sizes are small, so we have no power to say anything about how the genetic architecture or adaptive architecture of a single gene. We cannot say anything. We need to average across all the genes, and then when we apply tests for significance, we can say that we must have more than 5 on average loci contributing to the expression pattern for these genes that change their mean in expression. That's what we can say. It's a bit moderate. Yes? We haven't looked at. We haven't looked at. I can't tell you, we haven't done this. Okay, so basically what I can tell you, there is a way by you estimating the number of contributing loci using experimental evolution. It's not very powerful. So now let's get to a two-genotype experiment. Can we get a better handle on the number of contributing loci when we look at the genes themselves? I'm sure you have seen, if you look at Pauline's poster, then you see a bit of an idea how these Manhattan plots look like that you get from experimental evolution from pooled individuals. This is super messy. And it's very, very difficult to determine on this Manhattan plot the number of contributing loci. And this is mainly because linkage blocks drives up many SNPs, and then even if it's just a single haplotype block increasing in frequency, on a Manhattan plot, it looks like hundreds of loci that are showing significance. So you cannot do this by looking at a Manhattan plot. So the question, can you do differently? And this is these two genotypes experiments that we have been doing. It's a completely change in mindset. It's not trying to take into account or get rid of linkage disequilibrium by taking a polymorphic population at the beginning, but it takes the other extreme and says we start only with two genotypes. Two inbred isofemale lines are being brought together and let them evolve. So we start with complete LD, and we expect LD to break up during the experiment. So what do we see here? This is an experiment where we run these two genotypes for 20 generations only. And so what you see here, this is the position on the chromosome, and on the y-axis, you see the frequency of genotype 1. So there's only two genotypes. We plot the frequency of genotype 1. So we call this O in this case. It's from Oregon. It's Drosophila strain. And so what you can see here, then, this is the starting frequency, the dotted line. And then you can see that the frequency changes. So what's quite surprising is that is a very strong frequency change that we see within 20 generations only. You can see that this is a frequency change of 50% in 20 generations. It's massively strong selection that we see there. Pardon?
Where are the centromeres again?
This is, let me see, this is somewhere in the middle, somewhere in the middle. Here, there's a centromere here, and this is a centromere. Yes, but this is adjusted, this is centimorgan, so, yeah. Anyway, so what we see, and the other what I find super interesting result compared to what I showed you before, where we had all the redundancy that the replicates were doing different things, with the two genotypes now, we see a super parallel response. Each of the line corresponds to one of 10 replicates. So these 10 replicates evolve super parallel and show the same strong selection response. So how can we use this selection response to estimate the number of loci? This can be a very simple-minded procedure. So we can say, well, maybe we think there is one selection target. So we place a selection target there, and then we can simulate it, and we can ask, does the simulated curve look similar to our empirical data? And I think we don't need a theoretician to basically tell us that this is not true. So what we do is then basically we put a second locus there, and we ask, does now the simulated data look to what we have in empirical data? And you still don't find a match. So we put 3 targets there, and we still see it's not matching. So we put another target there. It gets closer, but there is still something here. Then we put basically 2 more targets. And now we say with 6 targets, it starts looking kind of similar to what we have in the empirical data. But still, there are still some very interesting differences that I like to highlight. So you see this is a very strong, pronounced peak that you see here. And this is a very broad shoulder that we have here. This is what we cannot replicate with this. So this suggests there may be here another target there that we haven't captured with this simple-minded idea. So how does it look like if we have now this experiment not run for 20 generations, but if we run it for 110 or 120 generations? So now each color is a different generation. And you can see this is the beginning of the experiment. This is a part of the X chromosome. And so what you can see is where we saw only a limited number of loci, now this chromosome is being broken up by additional recombination events. And this is always, I don't know whether you have, this is always the frequency of strain 1. So if the frequency of strain 1 is going down, this means there has been a beneficial allele on strain 2, there has been recombination, it has recombined back on, has recombined 2 breakpoints, and then this recombined haplotype is increasing in frequency, and that's why on this plot it is going down. So now, in principle, you could estimate the number of targets by just counting this here. And so this may sound like a very simple exercise, but I tell you, we are struggling with this. And I can't tell you really how many targets we have, because this sounds like a very simple task, but it is not, at least not for us. And so just to give you an idea how similar the replicates are now looking over 120 generations, and you still see that they look surprisingly similar across the 4 replicates that I'm showing you. So this the high similarity across replicates is persistent, but you can still see some very specific changes. For example, if you look here, this is a very pronounced peak. It's not so pronounced here. It's there, and there it's also only very weak. But if you go through this, you see that the selection signatures are being preserved. And so we are currently struggling, and if somebody has a good idea, I'm very curious to hear, how could we integrate the time series data and the replicates to get a good handle on the number of loci that are present in these 2 founder genotypes?
So if you're in a simulation with neutrality,
Mm-hmm.
you're still going to get some peaks and valleys by random chance. Now you're starting to see what you get if no loci are being targeted, simply getting separation.
You get much, it looks very unspectacular if you do.
On average, or any given realization?
If you have only 20 generations, that's what we've done intensively, you find basically nothing. That's it. The population size is not very small. It's still 1,200. 420 generations, you're right. 420 generations, you find some neutral, even on the neutrality, big changes.
So one thing to think about is like a bit more talk about. So maybe see that we basically moving along with random probability change. You can then estimate the number of change points.
Let's talk about this in. Okay, so now I've shown you, yeah? Yes, I don't have a problem with this. I think it's expected that they're more parallel than the other experiments where we start with many lines, because exactly what you say, there's not as much LD, and LD is one of the drivers for parallelism, of course. No, I don't have a problem. I just think it's cool. Yeah, this is exactly one thing that we want to address in the SFB, that we want to basically use this parallelism and look at advanced generations to see whether we get different breakpoints in the replicates and how is the response doing this. But I think what we need is a larger population size than we have for this one, because drift is still too big. It generates, they look surprisingly parallel, but if for this to be addressed, it's still too noisy. So we need to ramp up population sizes to be more confident on this. Well, it's 1,200 something. Well, I would need to know the architecture to do it really reliably. That's the point. And it will get me a different answer when I have many targets or few targets. So I'm.
So it kind of paraphrase me to kind of recombination. So it may be tricky to do. I know, for example, some variants of the biotic genes in software jump up from recombination. So if the curriculum goes in, then you get a lot more separation. Maybe the blocks will be small to go through the
That's another approach one can take, and some people have this in mind. But yeah, I find it easier for me now to increase population size than to, and don't know what the side effect of this could be. Yeah, but anyway, so let's go to the complications. First one is pleiotropy. And so can we get a handle on the impact of pleiotropy on our selection response? That's the question. How does pleiotropy affect the response to selection for these targets? And I always keep in mind how difficult it is to identify individual targets. So this is again the one of the experiments that we have from Florida, where we have 10 replicates that have evolved for 130 generations in a hot environment. So this is just to remind you that we have genetic redundancy, that the replicates are doing different things in this population. So our new test for identifying the action of pleiotropy is based exactly on polygenic adaptation and the high redundancy that we are observing. And that this means that different genes contribute to adaptation in the different replicates. So now, what this test involves, we mix replicate populations that have already approached the new trait optimum. And so they have done it by using different combinations of genes, and the different combinations of genes come with different pleiotropic effects. So what we basically expect that the mixing doesn't do anything because both populations have reached trait optimum. So the position of relative to the trait optimum is not changing, so we shouldn't find an adaptive response. They should not change frequency because they're still adapting. And if we mix these populations in an imbalanced way, this means we use maybe 15% of population 1 and 85% of the other population. What happens is that the alleles occur at different frequency. So the recipient population with the 85% will have alleles being present at a high frequency, versus the 15% immigrant. These alleles, the adaptive alleles, these alleles that were adaptive in the immigrant population will be a low frequency in the mix. Does everybody can follow me? So we have different frequencies of the adaptive alleles. And as a consequence, if these alleles are not fully dominant, they will be differently masked, because the low frequency alleles are more likely to be masked because they're more likely to be heterozygous than the others. So they're more likely to be masked by one of the other alleles from the other population. And as a consequence, if there are pleiotropic effects, negative pleiotropic effects, then these loci will be released from the cost of pleiotropy. And therefore, despite we don't expect any frequency change because of adaptation anymore, you may expect that these alleles are changing frequency because of the relaxation of the pleiotropic costs. Yes? Let's look at what we have and then we discuss again, yeah? You had also something, Enrique? Of course, differences in effect size also complicate the story. And I don't even know which allele. I cannot map positive alleles. That's what you have to keep in mind. So I will never know whether I talk about the real allele. So what we did, we basically took immigrant genotypes and recipient genotypes. These were 4 different replicates. This was a replicate that was very different in terms of the responding haplotypes, and this was one that was more similar to those ones. So that we had a gradient where was a one mixture that was very different, and one mixture was more similar, also creating different expectations. And then we evolved them for basically 30 generations, and you ask what we're getting. So the first prediction is that applies to alleles selected in the immigrant population, because these are the alleles that are selected in the population that has a low frequency. These are the alleles that are expected to be masked, and therefore we expect that they should increase in frequency in the mixture. So what do we see? This is basically the 4 different admixture events that we are doing. Each of the dots is one of the replicate populations that we have been running. And this is the frequency change that we see per generation. In red, you see the frequency change in the admixed population, and this is the frequency change in the immigrant and the recipient population. So you see that the immigrant and recipient population are not changing very much in frequency. But here, as predicted, we see a pronounced frequency increase, which is highly parallel across all the replicates. And it's very important that you keep in mind, we use all the alleles in the experiment and we polarize them according to their adaptive response in the immigrant population. So that's very important. We look at those alleles that are increasing in the immigrant population. And that's what we see. These alleles are basically increasing in frequency. Exactly. And so this is, you see that this is only happening in the first 20 generations. Later on, this response is already much weaker. So this is a quick response. And you can see this trend continues here again. You see basically the same thing. This is where we have the population that's less differentiated. And we see that the response is less pronounced, but it's still there. So it's a very, very consistent response. Yeah. But there's a slight reduction, but overall, I would say they're not changing in frequency. Here. Yeah, there's a slight reduction, yeah. But this is in the unmixed populations, yeah. It should not be polarized. The effect should not depend on whether they are in the alleles that are selected in the immigrant population or in the recipient one. That's the point. Let me see. So if you look at the allele frequency change in the recipient population, your expectation would just be the opposite. And this is exactly what we see. Here we see that they are basically going down in frequency because they are now still being exposed to the cost of pleiotropy. they're not needed because the other ones are going up. So they can go down, and these ones can go down. So the ones that from the immigrant population are going up in frequency, and so these ones can go down in frequency. And this is what we're seeing here. And this is, again, a very consistent pattern. There are no error bars. This is genome-wide. The error bars are massive. This is just average. So you can argue that this may be driven by recessive deleterious alleles, but the X chromosome, due to its homozygosity, doesn't have many recessive deleterious alleles. So the question is, if it would be recessive deleterious alleles driving it, you shouldn't see it on the X chromosome. And we see exactly the same pattern on the X chromosome. So I don't think that this is recessive deleterious alleles driving it. Another way to look at how these populations are evolving is on the PCA. So here we have the immigrant population, we have the recipient ones, and then you see basically at the F1, this is one of the immigrant populations, these are the two others, and they all move basically towards the immigrant population, consistent with the idea that alleles from the immigrant populations are going up in frequency, so they become more like the immigrant population. So the second prediction is that the selection advantage of alleles favored in the immigrant populations depends on the frequency of the hybrid population. Because if you have a low frequency, you're more likely to be heterozygous than when you have a high frequency. And so we predict that this effect should be stronger in low frequency alleles, or alleles that have a low frequency in the hybrid population compared to alleles that have a high frequency in the population. Again, the polarization is for alleles that are going up in frequency in the immigrant population. So what we see here, these are different frequency bins, 10% intervals. So this is the low frequency, this is the high frequency. Each dot is a replicate, and so you can see very nicely that the largest allele frequency change we see for the low frequency alleles, and as soon as the frequency goes up, the frequency change reduces, and then it goes even in the other direction. And this doesn't say that they're wrong, but it means they have a high frequency of 80%. This means that the same allele has been going up in the not just in the immigrant population, but also in the recipient one. And that's why it has a high frequency in the hybrid population. So these are alleles that are not restricted to be selected in the immigrant population, and they're very bad anyway. And you see again the same pattern that is highly conserved across all these 4 experiments. So it's a very, very general pattern that you see. So I hope I have convinced you that this pattern is suggesting that there is a cost of pleiotropy in our experiment. And this cost of pleiotropy, of course, is weaker than what we basically at the beginning, but you can unmask it by making this hybrid population. But the question is pleiotropy always bad? Is pleiotropy always negative pleiotropy, or should we think that pleiotropy could be also something good? Could we have synergistic pleiotropy as well? And I would like to introduce another study where we basically address this issue. This is a comparison of an experimental evolution study that we've been carrying out in the same regime as before, but it's now done for 2 different species. This is Drosophila melanogaster and Drosophila similans. They all have been collected at the same time, at the same location. So they really come from the same environment, and they have been basically selected in a hot selection regime and a cold selection regime. And then we were measuring gene expression. We were looking at the changes in gene expression in one species and the other species. And we were comparing the expression change to a natural population that is also has been collected in the north and the south. And the idea is the north is our cold regime, the south is our hot regime. And the question is, what are the similarities we're seeing there? So my question for you is, what would you expect? Is pleiotropy, what is the effect of pleiotropy? We have here a very simple lab environment where there's not much side effect if a beneficial mutation goes up, despite I've just shown you the opposite. If an allele goes up, but the environment is so simple that it could be that there's very little pleiotropic cost to this. But maybe the pleiotropic costs are much larger in the natural environment, because if you adapt to temperature, you also have to consider humidity, available food, predators, and all other factors. So it may be much more difficult in a natural environment to avoid these pleiotropic effects. So my working hypothesis was that we should find less influence of pleiotropy here than we should find there. So how did we address this? We basically measured the pleiotropy of genes with differences in gene expression. So what we have again we have melanogaster in, how should I say, this is the natural population, declinal population, and this is the experiment. And then we're measuring the average pleiotropy. So we don't classify them by pleiotropy, but we basically measure the average pleiotropy, the average pleiotropy. This is the distribution of the genes that we have here. And then you can see that the genes that are changing significantly here have a higher level of pleiotropy than we see in the experiment. And the same pattern we see in the other natural population similants. Here is the experimental ones. So we find that the genes with a significant change, I was expecting exactly the opposite pattern. I was expecting that the experimental populations, because the genes can respond, pleiotropy doesn't matter so much, will have a higher level of pleiotropy than those ones that we find in the natural population. So what is our interpretation of this result is that maybe it's wrong to overemphasize the negative effects of pleiotropy. The interpretation of the data could be that it's not negative pleiotropy dominating the response, but it's in fact positive pleiotropy. Synergistic effects, and they have been in these populations segregating, and these populations have been filtered out for synergistic effects. And therefore, in natural populations, what is dominating are the synergistic effects of pleiotropy. And therefore, these genes where we have synergistic effects are responding more, and that's why we have a stronger response here. So this is by no means conclusive. This is a suggestive interpretation of the results, but it may suggest that we have to also consider synergistic pleiotropy as a major driver for adaptation. Yeah? That's a good question. So we have used two different measures. One is connectivity, and the other one is tissue specificity. These are two different ways we used. And I have to say, we've taken these data from melanogaster, not from similants, but they're quite similar. It's of course, we've been criticized by reviewers by using these two measures, but I don't know a better one. That's the problem. So synergistic pleiotropy may be more common than at least I would have thought. Okay, let's go to the second complication, and this is linkage disequilibrium. So to start with, we said that polygenic adaptation means that an individual locus has a weak effect. But I've shown you data where we see frequency changes of 50%. So you can argue, well, there's a major effect loci that we have and has nothing to do with polygenic adaptation. But what could be the other explanation why we see these very pronounced effects? Why do we see frequency changes of 50%? And of course, I think it may have to do with linkage disequilibrium. So if we think we have linkage equilibrium, this would be all loci here with plus and minuses. And if we have free recombination, then basically you could have a situation that when you sample enough, all your chromosomes will have the same number of pluses and minuses, and there is no fitness difference. You don't find any selection signature. And if you basically have full linkage, then it's the same situation. There's no recombination. You have one chromosome with a certain number of pluses and minuses. The other one has another set of plus and minuses, but the net effect is the same. So it will be very difficult to find something. Here you have the full haplotypes. Here you have individual loci responding to a tiny amount. So you cannot detect any selection signature here, no selection signature there. So doesn't explain why we find it, but I think the explanation is that we have an intermediate situation, that we have some intermediate levels of LD in our experiment, where we will have some regions where there's a big difference between the haplotype blocks that we're having, with one block having a lot of pluses relative to the other block, and therefore then we have the combined effects of those ones resulting in a very strong selection, despite each of the individual loci has only minor effects. So it's a sampling effect, basically, that when you have intermediate levels of LD, that you should find a region that is responding strongly to selection. So to show you what is the impact of linkage, in our experiment, I should introduce another experiment to you. This is an experiment that we have been setting up, where we had a total 1,200 isofemia lines being collected from the same natural population in South Africa. And we divided these isofemia lines into 3 different sets. So each of the sets is coming from the same population, and they were randomly assigned. And then from each of these 400 isofemia lines, we graded 5 replicate populations. They're called group A, B, and C. So in total, there are 15 replicates. These 15 replicates have been evolving in the same environment for, I don't know, whatever, 80 generations. Good. So what do you expect in terms of the response? If you have the PCA that I showed you before, what would be your expectation? I have a natural population here. And I have 400 isofemia lines that come from the same population, other 400, another 400. These are different isofemia lines, these are different isofemia lines, these are different, but they come from the same population. So if I make a PCA plot, as I've done before, what would you expect? Altogether. Or I'll phrase it differently. Is there a difference between group A and B and C? Would you expect this? Well, of course, there's some sampling property that very rare ones will be missed. Yeah, that's good. So you expect differences between the A, B, and C. This is right? I would not have expected, but you're entirely right. So this is what we see here. This are the 3 different founders, so they're not very different. And you can see that they basically do entirely different things. Just to remind you, this was only one subset that I showed you before as evidence for redundancy. It doesn't mean that there is no redundancy. It just shows you redundancy is really of minor importance compared to what we have here. And, and the allele frequencies are not very different between them. And this is something that builds up over time. And the explanation is that this has to do with linkage disequilibrium. And linkage disequilibrium, or haplotypes, has a much higher sampling variance than allele frequencies. because you're doing very well by sampling only 400 isophemal lines, means in this context something like 800 genotypes. With this, you can estimate allele frequencies quite well with relatively low sampling variance. But if you think that alleles are recombining, and then you're not sampling very well the haplotypes from this population. In fact, you have a huge sampling variation which of the haplotypes you're selecting. And so it's the haplotypes that go into the founders that are determining your selection response.
Your linkage for, I don't know, 50 generations and you run experiment. There you would expect that this will look better. Is that correct?
So, no, that's not what we did. I should say also we run simulations by asking a question, how large does your sample size need to be that you don't see this effect? And of course, what we find is that the larger your sample is, the effect will be smaller, but even with a sample size of 10,000, you still see the phenomenon. No, isophemal lines.
Okay.
Basically, this is, so the idea is that it's the sampling variance in terms of the haplotypes that you sample, which is determining the outcome of your selection. And this means it cannot be representative. What you do if you go out, you take a sample from a population, this is not representative if you evolve this, because it was just basically contingency, which haplotypes you picked up, what response you're getting. And this I find quite sobering, but yeah. We simulated it, and it's when we have this effect, we run the simulations conditional on having the same alleles, we still find the same phenomenon. So you don't need this. This can also, in addition, contribute to this, but it's not the major driver. So we see the same thing when we can do it with simulations only, where we basically generate different haplotypes, and then we still see the same phenomenon.
How rare are the rarest alleles?
I can't say. I don't think it's the frequency of the alleles that matter so much. Because it's the frequency is already quite low. Could be.
Then the stochasticity of losing them quite quickly is quite likely to be losing them quite quickly.
If they're the very low frequency ones, yes, you lose them quickly. That's true, but the simulations show that when you don't have these ones, you still see the same phenomenon because of the LD patterns. The LD patterns are driving this different response.
So instead of rare alleles, you now have rare haplotypes.
Exactly, I think.
Those rare haplotypes have a large effect. They can increase in frequency dramatically.
That's what I think. It's not with 400, you think with 400 individuals, you capture the frequency in the population very well, which is true, but you don't capture the haplotype distribution very well, and this gives you a different response. So that's why I think it's very important to keep this in mind, how it matters. I should also say, the effect is actually stronger when you have a weak, when you have few contributing loci than when you have more. The more contributing loci you have, the weaker is the effect. So. We have done it at some point, and we found similarities across chromosome arms, but we haven't done further. But it's maybe a good idea to see how it looks like, yes.
So one way to think about it is you have a haplotype. And let's imagine you've got equal numbers of plus and minus. So if there's a small number of slots there, then your variance, think simple limit here, right? You can have a larger variance in terms of plus minus sum across those. As your number of plus or minus gets larger and larger and larger, they tend to center on that one value. So fewer ones, fewer sites would be basically more variance in fitness.
Yeah, that's, I should also, in response to, we looked at this pattern when we excluded, but it's not a full answer. When we look only at the common alleles, it's still there, yeah. But it could still be that the few ones are the drivers. But if you look at only common alleles, it's the same pattern. Possibly, yes. I would think so. It would greatly enhance it, I would think. Yeah, yeah. Good, so now let's look at the third complicator, and this is the multiple stressors or E by E interaction. So just what I mean by E by E interaction, it means that, well, there is some data out there that when you put a population to one stressor and the other stressor, if you combine these two stresses, you get a different response. You cannot predict the phenotypic response of a population by looking at the individual stresses alone. So this is it's somehow characterized on the phenotypic level. We do not know much more about this on the genomic level. That's what I would like to discuss. So we basically, it's our typical setting. We have the natural population, and we bring the flies to the lab. And the natural population, they differ basically for many factors. It's temperature, food, sexual selection, reproduction strategies, whatever, name it. There are many different phenotypes that these populations have to adapt to when they're in the lab. And so the question is, how can we look at the environment by environment interaction? The way that Zhangyi came up with looking at this, he said, well, we could keep everything constant, but we modify only one factor. And this is why what he did, he modified only temperature. So he basically had the population set up in the same way, they were brought to the same environment and everything. The only thing differed was the temperature. He had one environment in which the temperature was a constant 23. In another environment was also, the average temperature was 23, but now it was fluctuating between 28 and 18. Apart from this, they were adapting to the same thing, and they have been evolving for whatever, more than 100 generations. Okay, so what would you expect in terms of the phenotype? And I don't know which phenotype to look at, so we better use fitness. So if you look at the fitness of the flies, this is the ancestral population, and these are the two temperature regimes that they have been evolving to. They have been assayed here, and this is for the constant temperature. So you can see that the ancestor, they're both significantly better than the ancestor, but you cannot distinguish them here. This is one measure of fitness, this is another measure of fitness. It's very consistent. You see the similar response when you put them in the other environment. So you typically find that there's very little difference between the two populations evolved at the two different temperature regimes, but they have massively increased in fitness relative to their ancestral population. So the conclusion is that the evolved populations have very similar fitness. And temperature regime doesn't have a major effect. So it doesn't matter very much whether they have been evolving in the constant temperature or whether the temperature has been fluctuating. They have been probably adapting to the other environmental factors to a much larger extent. Okay, so how does it look on the genomic level? What would be your prediction here? Well, I would have expected, based on this, a parallel response. I would have expected similar alleles to change in frequency there. Of course, there's all the problem of redundancy and all this. So what do we find here? These are these PCA plots that you see here. So what do you find? There's a clear divide. On the one side, we see the ones in the constant environment, and here we see the ones in the fluctuating environment. They're super well separated from each other. We see still redundancy. There is redundancy, but redundancy doesn't explain as much of the variation as does the temperature regime. So on the genomic basis, we see a distinction between the temperature regimes, but we do not see it on the fitness level. So can we look a bit more in the genetic response? Because you may say, well, the PCA is too superficial. Now we do it on a SNP by SNP level. You see this is the number of SNPs that are overlapping. If you look at the correlation allele frequencies, you see that within the same regime, the allele frequencies are quite correlated, or allele frequency changes are quite correlated. Here as well, between the regimes, they're not correlated. And if you look at the SNPs individually, so there's SNPs that are selected in the constant environment. You can see they have quite big frequency changes here. If you look at the same SNPs in the other environment, you see that they're basically not changing. And the other way around, these are SNPs selected in the fluctuating environment. Again, we see big changes, but if we look at the same SNPs in the other temperature regime, we see those ones are not changing. So this is all consistent with a highly specific response, selection response in the according to the temperature regime, despite they seem to be adapting to the other common stressors to the same extent.
Phenotypes are just making them do fitness, but it's not necessarily this, that they are adapting to something else. They are adapting to the actual environment via different genetic changes because they are environment-dependent adaptation. And it's just these changes in, for example, I don't know, hydrocarbon physical, depending on temperature, make them more fit.
Sorry, I don't get it.
Just fitness is fitness.
Mm-hmm.
The actual trait that you don't know what it is. Might be differential between the populations.
But why would then the fitness be the same? Yeah, but why do they do it with different genes?
Because the phenotype you need to adapt, the phenotype we're not measuring, but you need to adapt to that environment. I'm just imagining that could be an alternative explanation, not necessarily that they are adapting to something else.
But the typical expectation would be that they would have basically different fitnesses in the non-native environment. You would expect, if you put them to the non-native environment, that they do worse. Where do I have it now? Sorry, if this then you would, this is in the constant environment, you should expect that the fluctuating should be worse. Because they're not adapted to this. This is not the home environment. This is swapped. Yeah, but it's not significant anyway. They're the same. And if you do it in the fluctuating environment, yes, they're slightly better than there. But so this is what you would expect. Of course, you're right. This is only one measure of a whatever high level, super high level. But I'd rather think my interpretation is that you basically cannot really predict the phenotypic response or the genetic response when you adapt to multiple stressors that are interacting. And this is, I think, the super interesting take-home message, because if you think what people are doing, there is a whole machinery of studies in molecular ecology where people are trying to associate genetic variants to different environmental variables. And so the assumption is that all these different environments are acting independently of each other. And so we have a very simple case where we show that this is not the case. But if you extrapolate to all these studies, this is a useless exercise, what you do. So I think this is, to me, the exciting take-home message, that the interactions of environments can create a super different selection response to this, which is, in my opinion, not being considered before. But so the last complication, yes. So I don't have to talk much about this because you all have seen Vidadi's poster. I just want to remind you that we have done an experiment with different starting frequencies on the X chromosome, but the identical genotypes, 2 genotypes, but they started with frequency of 30% and 70%. And then when we calculate the selection coefficient all over the chromosome, we see that they differ. The selection coefficient should be independent of frequency, so we would have expected they are the same. And Vidadish has an explanation for this. He thinks it's epistasis driving this. I hope you've seen his poster and he convinced you. So I will show you more data trying to convince you that this is the right interpretation. So you can do another 2 genotype experiments with 3 genotypes. You have genotype A, B, and C, and you make the experiment with A and B, with B and C. And if it's epistasis, which is Vidadish's preferred hypothesis, you should find a deviation from the prediction from AB and BC comparison compared to the observation of A versus C. Whereas put in other words, if it's purely additive, you can predict the response of the AC genotype by combining the response from AB and BC. And this is what we have been done here. And I was focusing on the difference of Samarkand, Oregon. This is the one that Vidadish has also on his poster. So what you find here is basically there's a dotted line. This is the observation. When we look at the differences, where we look at the selection response that we observe for the Samarkand and Oregon experiment, and the solid line is the predicted selection response, and predicted based on the selection response of Helsinki, Oregon and Helsinki, Samarkand. So this is the prediction based on the competition of two different genotype combinations. And so what you see here on the comparison of the dotted and solid lines is that on the autosomes, there are some differences here that are consistent. So there may be something here on 3R, but the major thing what you see here is actually on the X chromosome. So you see exactly the same pattern that, that he saw, where we have the frequency increase here and it goes down, but the predicted response would be entirely different when we use these 2 separate experiments. And so the conclusion is that this has nothing to do, it's basically additive effects cannot explain the response in these 2 genotype experiments. And as Vidadish has shown that it cannot be explained by dominance, and therefore what we think is left over is the explanation that is epistasis driving these differences. And this is a, to me, quite stunning result, because again, I was thinking that I always have the wrong intuition, that it's basically additive effects for adaptation would be more important than epistasis. But here it shows that epistasis is very, very important for determining the outcome of the result. Of course, these are 2 genotypes which start with similar frequencies, and therefore the influence of epistasis is stronger than you would expect if they would have very small frequencies, but it's still a very surprising result. So when I finish off.
So this is substantially more epistasis than people have
nobody knows how important epistasis for adaptation is in sexual species.
That's some of my colleagues, when I asked them, said, oh, it's very weak epistasis.
Yeah, well, this is from GWAS.
Yeah.
This is from GWAS, and the difference is probably it has to do with the frequencies. Here we start with frequency of 50-50. And so that's where we maximize.
Much more sensitive measure.
Exactly, I think so. But you can see the difference is quite substantial. This is the prediction on additive effects, and this is what we see here. This is massive. It is massive. So epistasis can be extremely powerful to influence selection.
But it's not just power, it's about the initial frequencies that have as opposed to sampling from the water.
Right, but I mean, if it's parents and you care about it, the frequency is No, it's the question of whether it's statistically significant in the population, and the question whether it's biologically significant, which has nothing to do with that. Right? The biological significance is mating between individuals and all the groups and so on.
Anyway, I was assuming that recombination is more screwing up the epistasis. But anyway. I should thank the people having contributed this. This is a snapshot of the lab. It's not very up to date because the 3 members that you see from our lab are not here. But anyway, so I'm happy to hear more suggestions from you, what we've been doing wrong, and thanks for your feedback.